Emily Foster. Yeah.
And then,
I didn't see Brian Pulmonary.
oh, Brian was there
I didn't see him.
at the bar.
Oh, that's a great place to be.
He's like, we're doing shots. I'm like, no, we're not.
Dr. Meyer.
Got that. Nina Delaney Foster Farthing. Oh, there's a new office there. Did you hear about that?
Is it the Avante office? Dr. Arsena, do you call her Palmer?
She's the one.
Just open. How do you spell it? Arseneau. She's been there for a while. Love her. She cranks out screams.
Really?
Yeah.
Well, I am in that
That's awesome. I'm gonna eat
And
that.
she has a new baby that she's got like a 7 or 8 month old little baby girl. So when you go into her office, there's like a whole charge kit like everywhere. It's so I love it. SENEAU. She's awesome.
How long have you been with AstraZeneca?
Almost a year.
Do you love it?
Yeah.
What's your territory?
So it goes far north as Snoqualmie, and then as far south as Canton. Everything in between.
You have a territory really similar to mine where it's like really small geography but super dense.
Yeah.
So I have at Ellensburg to like all the way to here to downtown, and then at Northgate down to like Auburn and Burien, and that's it. So
Okay.
you just
All the way to Ellensburg. Wow.
there's like 2 targets out there. It's not, and I live in Snoqualmie, so
Okay.
it's not hard to what.
Sticking targets the shop.
What?
Target.
Oh, the store. No, there are not 2 targets in Ellensburg. If there were, that would be awesome. Yeah, so it's not hard. Like I can go there and back in a day, and there's not, it's mostly just kind of use.
What about when it gets snowy though? Oh, like
Yeah, I
that
probably
tricky?
wouldn't.
Okay.
I'll just watch it and see if it's what it looks like, and
Yeah.
and I can always mail stuff like if they needed copay credits or whatever. Yeah.
Oh.
That's nice. I kind of small territories you can bang out calls really quick because they're like all together.
Yeah.
That's fun.
Did you get a number of other people to cooperate?
Well, it's still the beginning. It's still early.
Okay.
So Shelly's all over it.
Next week.
And then
I'll be bringing breakfast as well.
Matt, you
Excel did.
And I might.
So
can Matt do it?
I think so. He just wasn't sure because the last time he said he spoke to you, you were
Oh, well, I've obviously broken that rule already.
going to. So I
Well,
like
this
it.
caterers, I think if we can all stick with this one, I mean, they were here early, which is,
They called me at 6:15 and they're like, "Where are you?" I'm around the corner.
yeah.
So
Yeah, I was glad I was here early because the speaker was lost.
Oh,
Just
yeah.
called me to get in the building.
There's a lot that needs to happen.
Yeah,
Yeah.
absolutely. I said, this is,
ARSEN.
well, they're recording everything we say from this point on, so I'd be quiet.
Perfect. Let's get a good lawyer. Okay, great. I need his name.
And Dr. Alvin.
Hello. Yeah, we bailed you out. Hi.
I remember you. Have a nice summer.
Just came back from California National Park.
Is that right?
Yeah, just took a short vacation. Ever been disappointed?
No, I haven't. The Kings Canyon and Square I haven't been to.
Good morning.
How are you?
Boy, I was excited.
Did you climb Mount Whitney? I don't know. It's in there.
I can't actually see it. It's hidden by the slightly shorter mouth.
Hey, the last time I saw you, I was got some wine for me to drink that stuff off.
Yes.
How are you? So I see your little Facebook post all the time.
I know, I'm always traveling.
You are, yeah.
Yeah.
It's gone. It's all gone. Going into fall, yeah.
Are you sure it's not too
We got this Wednesday golfing from our men's senior group.
Oh, you still got it.
I started dancing back. So I got a few things going on. Cooking classes, you know. Trying to stay out of trouble.
Well, I think it's pretty loaded already.
Yeah, I see the presentation, but for the.
Well,
I'm.
Well, Drew, one of our young faculty.
Should we have a lunch date?
Yeah.
We
got a nice
I'm
tan.
so sad without you there. I mean, we have everyone else, but we miss you.
Your trip okay?
Yeah, I missed the whole, but what the heck?
Taken last night.
Good for you.
Kind of live a little, huh?
I've got.
Are you staying out of trouble?
Mostly.
Hi, Dr. Ayers.
You've had an interesting summer, haven't you?
We checked in to make sure, like, should we sit at the end or should we?
Are we gonna get kicked out or what's the deal?
You showed courage. That's a good sign.
Okay.
I'm about 40 to 50 minutes, but I
Oh, you'll be
can should we come? Yeah. Oh,
Oh
my, here's hoping.
With our jobs intact.
You know.
Outside audience as well.
Horizon? So there was one of the Horizon reps with you last year. What a nice guy. Yeah.
I'm the medical director.
Oh.
That's the way to travel. There's no parking anywhere.
Wanted to come and
Can you find a place for your bike?
Yeah.
So you might be just back.
So I can feel your phone.
And in case I forgot which building it is, I got lost.
Well, we've moved from that building
Yeah.
about a year ago to this one.
Great breath. Interesting.
You guys get back.
You took a slightly earlier flight, so we got back about 3.
Yeah.
Oh, yeah. I just showed Gary, I was just telling him we were there. It's actually a sequoia tree. Biggest tree in the world.
General Sherman?
It's not the tallest.
Oh, no, I
it's
can-
big,
Okay.
but it's the biggest.
You gotta have a picture of yourself.
I didn't see any around here. Usually I would see like Starbucks.
Museum.
There's actually
You went up to the floor. Almost have really valuable art here because you have a garden. And I came to eat wax.
I've seen the pictures of General Sherman.
They helped me last night. Taking care of it. And I said, I'll have to work on my face.
No, yeah, well, they're big enough. No, I wasn't. Where did you fly into? Your Carmel? There it is. That's
And this time of year, there's no traffic.
It's on my bucket list. Yeah.
I looked at all the signs, said no parking, it's from 9 to 10.
Yeah, that's,
Sold in the China shop.
someone's gonna remove those signs, because it's deceptive, you can park there.
Paul, when are you gonna retire?
The Viva thing, I had to sign a 3-year contract extension. So I'll be there for at least 3 years. I don't know if I'll be full
How
time.
about you? Yeah, same loop.
When Kaiser takes over, there's a whole.
Corridors, yeah.
You gotta be there.
Clearly.
Oh, I see, yeah. Okay, so we got motivation to carry it at the end of the stick.
Wow.
Well, what's keeping me going is the
That's
lease. I got an expensive lease. I guess I'd get somebody to take over.
On the building thing?
Yeah.
That's probably manageable.
Oh, actually, that's my rationalization. I just at least put my dad in another piece.
I know it's quite
Something that's
well.
easy to do.
Yeah.
Good to go.
So just let me know if you have.
A lot of motivation. I think it'd be a lot harder to work more than I can.
Yeah, I haven't had a patient in the hospital in over a decade, probably.
Yeah, whenever you get.
Hospitalist.
There's no rush.
Pulmonary,
But we'll
they
be,
say
we'll
early
be
surgery.
on call this
I'm more than happy to.
So, fabulous.
Yeah.
Yeah, they just.
Got any ski trips planned this winter?
May
I don't have any hip replaced this summer.
Oh, did you? What?
Probably will ski, but probably be.
What, the intermediate slopes, yeah, right. How's that hip working?
It's much better than what we did.
Hurts before.
No, we're gonna start doing hip transplants. Bring people in at 7 in the morning, they're gonna operate on them, send them home at 10
Now,
at
you
night.
said hip transplant.
Implant.
Oh. You know, that'd be new. Take one of his hips when I was
Every
old.
time I saw one, I'm like, oh no, and then it was like a
No, you can
a worker.
refurbish them.
It's amazing what they're gonna get.
Yeah, that's quick.
Okay.
I can see that, you know,
The reason why.
first day, one full day.
They're talking about doing it as an outpatient. It used to be in the hospital for like 2 weeks. All these machines on your knees and stuff. Boot straps.
You're gonna have to.
I was
Before the
like,
surgery.
all parents were like,
Well, now that they're using the ones without glue, they're just hammering them into your toes.
Hopefully all the cheese
Well, it's 2 years studying, so.
You hope they will. Did you go home on blood thinner? You go.
But there's a lot of objectives.
That's it?
That's the thing. We've been having a lot of these people, we swear up and down that they have this extra.
Well, they got you on the move.
Yeah. Same goes the provocative use, plus one is needed. So they don't have to go close to where they were.
They're actually doing 2 at a time now.
And so they're just making this up. I don't know what it is, but
They're really fine.
you see physiological changes. Negative exploration. You can see them like go like this.
These
People, their next line, they have changed from this.
I cannot, I am so sorry. The pen? Yeah.
I had my shoulder done a couple of years ago. The hardest part was not being and being able to only sleep in one
We
position.
had a couple of perfect ones.
I had to rotate a couple and tore my biceps tendon, so they surgery all over. But you're locked like this, so you're not allowed to move at all. And so the only way to sleep is on your back sitting down. And you get so tired, I can't go one side, I can't go the other. No, I
They
just.
give you some drugs to at least knock it out.
They gave me, I had drugs, I didn't really need it for the pain, but I needed it for my sleep.
And not because you
But
go
I go
down.
stir crazy, that was the worst part. And I'd wake up at 2 o'clock.
Yeah. I don't know, nobody asked me.
I
Maybe
told
Frank?
Tina, hopefully Frank will.
And nobody stopped by and grabbed me.
Yeah, don't worry about it.
Other thing about my shoulder, you know, I'm right-handed. So my right shoulder.
The drug itself.
There's some tricky business there. When you can't use your right hand. Eating these tough and liver issues too.
Components of the
Yeah.
the sham has the same other stuff without the drug.
I had
Yeah.
I lost use of my hands. I had a carpal tunnel and I had a whole new
Put them into like
at the same time.
Trendelenburg type
So they did both surgeries at once. And I could Sue, can you come help me? I don't really want to. That's what you want a bidet.
You adapt, but it's still very clumsy. You realize how dominant hands you are. Eating is tricky.
I'm trying to button buttons. You're like fumbling around, can't get anything to work.
I can do my job. My hands like this and swing. Right. You know, it's like that. Allergists, we aren't surgeons. We don't need to use our hands at all. And that, which was great. That was, work was the best distraction of all.
And then
I missed 2 days of work.
Is Kevin still with you?
don't know
No,
why.
Can we come in a journal?
Yeah, he quit. I don't
Tickets
know.
you have.
You know, I got none.
None?
Well, I got 4 tickets. I gave up 2 because Drew would know the other games. Drew needs his father. Now he has to take care of his kids.
So they're all being used.
Yeah, and those are banned right now. It's going to be a sellout. No, that's true.
But it's the next week.
How much of your time?
My only problem is I don't have any extra tickets. On the other hand, we're going to game with all the games on the boat. You just come on and hang out on the boat, have fun, go to the game.
And then you just watch from the.
Well, you can have your stadium. What happens is you sit on the boat. Some of the games will be televised. You can watch it on TV in the boat. But the other thing is just the whole ambience. It's a big tailgating party down there with all the people in boats, and half the people don't go to the game. Some of them have big TVs, screen TVs, and watch it out there. But it's just fun. People drinking and having a good time. It's a beautiful Sunday game.
No, I'm fine. No, I'm fine now.
It's a Friday night game starts at 6.
Let me tell, so you're definitely going to do it that way.
Yes.
Let me tell Catherine.
You could meet down at the other option, probably the easiest, because we take the boat out of Kenmore, the very north end of Lake Washington. You can meet at the boat. Crew house. Meet there.
So I do I mean, I missed.
How about you, Drew? You staying out of trouble?
He had
I have no time to get in trouble with 2 kids.
He has another kid, May 22nd. Your grandpa yet?
Yeah, I have one grandson that lives in Virginia. He's going to be 3. I get to see him about 3 times a year.
Not that far away, that's pretty good.
We do like the part, seriously the part.
So you,
I mean, I got here.
$500 each.
what was the word of the day?
Like my whole
This is what I was waiting for your answer.
What was the word of the day?
Signaling,
Signaling.
whatever. Must have something to do with archive.
Good guess.
I don't.
No, even with no glasses.
Is it?
How are you, Nick?
He's alive.
He's alive.
Still.
I can read the title, Gary, and I can make out some of this.
I can, but my brother got squint.
Maybe we should make a really good PowerPoint on the race.
Yeah, for us old guys. I'll tell you, even the young guys. You know, the slides have gotten so much more complicated over the years. Just because the science has gotten so much more complicated.
Where'd you get your?
Back to you.
Sitting next to you.
So, like, the new ones are right.
Yeah, so she's from our early development.
Go on to the next one.
Take the seat, let's go ahead and get started. So, welcome to the beginning of our 2016-17 season. Somebody asked me, Zoe asked me on the way in how many years I've been doing this, and I just said to her, more years than you've been on this planet. Hopefully everybody got the schedule, and I haven't had anybody reject the assignments I gave them, so I'm assuming you know what you have to do, and you'll do them in a timely way. Just a couple of announcements before we get started. This is a picture of Seymour Klebanov. Not too many people in the room knew Seymour. Frank, Bill, Gary, myself, Mike Kennedy for sure. Seymour was the head of the Division of Allergy and Infectious Disease and a mentor to at least those I mentioned and many others who aren't here. He died a couple of weeks ago at age 89. He was a world-leading scientist. He was an MD and a PhD, but to my knowledge, he never really functioned clinically as an MD. He was really a PhD. One of the world's leading people in the in white cell biology and specifically in chronic granulomatous disease. Certainly taught me what little I know about immunodeficiency from an earlier era when that was not such a complex subject as it is today. So, a lovely man. I don't know, he was obviously getting honored the way he was dressed here. He had many honors if you read about his life and his history. The other things I just wanted to do this morning were introduce to our wider community, ourselves here and people watching from the outside, the two new fellows. I want you guys to just come up here for a second and say one sentence about who you are. Jay, you want to come up here?
Hi everyone, I'm Jay Patel. I'm one of the new Allergy Immunology fellows at UW. I'm from LA originally, and I did medical school out in Philadelphia at Drexel, and then I did my pediatrics residency in Children's LA, so I'm glad to be here.
Christina?
Hi everyone, I'm Christina Swanson. I grew up in Portland, Oregon. Did my undergrad here at UW and then my MD PhD at the University of Colorado. And I did children's training as well at OHSU at Dornbecker.
So you're a duck, sort of.
No, she went to
undergrad. But you're both, right?
And a buffalo.
So we have a new faculty member who, frankly, I wouldn't recognize if she was in the room, but I don't think she is. Bill, no? Larry Rampour has joined the faculty. Bill, you want to say anything?
She'll join faculty in middle of October. Assistant professor, finished her AI fellowship at Einstein. So very excited to have
So she's
her.
in town, but not started working yet as faculty. All right.
What was her first name?
Lahari, Lahari, and then Rampur, Rampur.
She's primarily going to be a clinician.
And also direct our didactics for the ABI board review. So that'll be a major thrust to a structured curriculum there.
All right, well, let's go ahead and get started. Thank you for giving me a couple of minutes of your presentation this morning. We have a guest speaker, kindly came up from Genentech. I hope I pronounced your name correctly, Dr. Pappu. A researcher at Genentech working and talking about epithelial-derived cytokines in pulmonary inflammation. So thank you very much for joining us.
where we just interrogate. Well, we identify pathways we think will be important in different diseases, and we interrogate whether we think these will be viable candidates or not. But it's very cellular, molecular, and animal model level. And just for disclosure, all of this work was done at Genentech, and we're all employees of that Genentech
Yeah.
and Roche.
Speak up. I'm
Okay,
gonna
sorry.
keep telling you that through the whole talk.
Okay. All right. So we know that we appreciate that asthma is a disease of the airways where there's various environmental challenges that can trigger inflammatory cascades that ultimately lead to what we see either eosinophil or neutrophil mediated inflammation, fibrosis, mucus secretion, and smooth muscle contraction and hyperplasia. And all of this, of course, leads to what you see in the clinic in severe asthma, which is exacerbations, airway obstruction, and airway hyperreactivity. What we're really interested in understanding is what are the molecular pathways that go from when you have that initial trigger from the environment to these downstream effects. And we've learned over the years that there are many leukocyte subsets, which I've highlighted here, that contribute to different aspects of the pathology, either through secretion of cytokines such as IL-5 or IL-13, chemokines, mast cell degranulation, etc. And what we've learned from the clinic is that agents that target these type 2 cytokines, such as IL-5 or IL-5 receptor, have shown efficacy in a subset of patients that display these elevated levels of type 2 biomarkers, such as eosinophils. We also learned from the clinic that there is a subset that doesn't seem to respond to this type 2 cytokine-directed therapy. And these display the biomarkers that are like decreased levels of eosinophils or periostin or phenol. And so what we're really interested in Genentech is to try to understand what's happening here upstream, where I have the arrow, and I apologize that I cannot seem to work here. This is very sensitive. Okay, so what are the upstream cytokines? And so it's been shown that the epithelium can contribute to these airway inflammation, and namely through secretion of or expression of cytokines such as IL-33, IL-25, and TSLP. And so a little bit about what these cytokines do. So we know from various works that IL-33 is actually constitutively expressed in stromal tissue, so it's not just restricted to the epithelium, but also you can detect it in fibroblasts as well as smooth muscle. And that cellular damage, either through some viral infection or an allergen chemical, leads to death of or damage of the epithelial or stromal cell and allows for release of IL-33. So if you can imagine, this is a very immediate process following exposure of your lung to these different environmental challenges. In contrast, it's been shown that IL-25 and TSLP actually do require a transcriptional regulation. So the epithelial cells receive signals from the environment, and that then leads to transcription and secretion of these two different proteins. And so you can see kinetically why we think IL-33 is probably more dominant, just because its expression seems to be not as delayed in regulation. So what do we know about what these cytokines do to these different cell types, which I've highlighted here? So we can detect expression of the IL-33 receptor, which is also called ST2, which I've highlighted here in pink, on different leukocyte subsets. And you can see here, what most of the subsets we think are important for asthma, such as mast cells or type 2 T cells, express the ST2 receptor. In contrast, we can only detect the IL-25 receptor on a subset of the leukocytes, such as Th2 cells, NKT, and then these innate lymphoid cells. And likewise, the TSLP receptor also seems to be much more restricted in expression compared to the IL-33 receptor. And so all of this data, suggested to us that IL-33 may be having a more dominant role in vivo in type 2 inflammation. And so that's going to be the focus of my discussion today. And so a little bit about IL-33 biology. This is once it's released, it can then bind to its receptor on different target cells, allowing for the formation of this complex, which is ST2 and IL-1 RACP, and then it activates the NF-kappa B MyD88 pathways. So you can see here this then leads to induction of different cytokines such as IL-4, 5, and 13. There is a receptor that is soluble, that's product of alternatively splicing of the IL-33 receptor that is thought to act as a decoy, which I've shown here in the purple line. So what really got us interested in IL-33, aside from this preliminary biology, was the identification of SNPs in this paper here in the New England Journal a few years ago, where they identified both SNPs and the IL-33 receptor, which is IL-1 RL1, and IL-33 itself, that associated with asthma risk. And you can see here what we did is we not only were able to replicate this in our own in-house studies, but we tried to see whether these SNPs associated with either the EOS high or the EOS low asthma. And what was to our surprise is we found that it associated with both, so there was no segregation according to biomarker. And then this was actually in contrast to what we found with other Th2 genes, such as GATA3, where we found they were only enriched in the EOS high or type 2 high cases. So in doing this analysis, we identified a set of amino acid changing SNPs that were in tight linkage disequilibrium with the original ST2, the IL-1 RL1 haplotype. So this block really is all inherited together. And so what we find here is that if you look at the odds ratios, they're less than 1, and so suggesting that this SNPs are protective. And so we wanted to understand what these SNPs might be doing in terms of IL-33 activity that would confer this protection from asthma risk.
Can I ask you a question?
Sure.
So if IL-33 is constitutively made and all those cells respond to it, all have receptors, you're talking about its disease potential here, but what's its biologic potential? Why would it be set up that way? It must have a beneficial function.
So we think it's actually tightly regulated. It is, yes, it's released upon damage of the cell, but we think it's really, that damage part is, we don't understand how it gets out of the cell and what's the actual signal that's the damage that releases. But we know from studies using transgenic mice that if you overexpress like a soluble IL-33 where it's no longer, it's actually bound in the nucleus normally, and where that nuclear localization is lost, and so it's constantly being allowed to get out, that those mice die of massive type 2 inflammation. So there's some regulation of IL-33 that we just don't get.
But I'm just here, the entire focus is thinking about it as causing disease.
Yeah.
That's not the way nature was designed.
Yeah, well, we think it has a role in anti-helminthic immunity. I'll show you some, at least from animal models, it seems to have a role in anti-helminthic immunity, as are most of these type 2 pathways, right? So a little bit about where these amino acid changes are. So if you can see here, two of the amino acid changes are in this TIR domain, which is an important domain for protein-protein interaction and MyD88 signaling. And the other two you can see are at the very C terminus of the ST2 receptor. And so we wanted to understand whether these amino acid changes, especially the ones in the TIR domain, were somehow affecting IL-33 activity. And so what we first did is we overlaid, we don't have a crystal structure for ST2, or at least the intracellular domain of ST2, so we overlaid the two SNPs in the TIR domain that onto a related structure, that of TLR10. And you can see here that the A433T mutation lies in a region that's really close to what's called the BB loops. And these BB loops are really important for the MyD88 signalosome assembly. So even though it's a subtle mutation, if you think A to T is really not that big a deal, we still think that because of the location of those mutations, they can still affect activity. And so to interrogate this, we used first a cell recorder system. So this is just the signaling of IL-1, which is a related cytokine to IL-33. And you can see IL-1, once it's added to cells, it activates NF-kappa B. You can use a reporter cell line and measure activity by the induction of the AP1 NF-kappa B transcription, which is shown here. And because IL-33 and IL-1 are pretty much identical, they just vary in which target cell they activate. And you can introduce ST2 into these cells that normally only signal in response to IL-1, and then by introducing ST2, you can see here that you can now confer IL-33 activity. And this is what we see when we have the common variant. So what happens?
Can you use the pointer on the screen?
Yeah.
See if you can get
I
that
can't
to work.
seem to see it.
Drew, can you
There you go.
Okay, now do you see it? Okay,
Yeah.
got it, okay. All right, so this is what you see with the common variant. Now what happens when we introduce the same receptor with those 4 amino acid changes? And the reason we did all 4 at once is, remember, this is all in tight LD, so it's always inherited together. And so what you can see here is in the blue that there is a shift in the EC50, so the IL-33 responsiveness, which is about 4-fold. You can see here that we've shown in this table right here. And then just as a positive control, we also looked at IL-1 and there was no change in activity. So we really think it is due to that receptor mutations and nothing to do with intracellular, something intracellular inherent in the pathway. Okay, so then we...
you said you did all 4 together. Those 4 mutations are not in linkage disequilibrium.
They are. The whole haplotype is in linkage disequilibrium.
But those are all within the same protein, right? You're saying. I thought you were saying that's in linkage disequilibrium with, because you had 2 SNPs on your GWAS.
So okay, so I can go back and show you, explain it. Sorry. Here. So the original Moffitt paper. See if I can find the pointer. Sorry. So the original Moffitt paper was this intronic SNP.
Right.
And then so we confirmed that in-house, and then we also identified in tight LD with the intronic snippet that was first identified these 4 amino acid changing mutations. So the whole block with that intronic SNP is in tight LD.
So you just had a higher coverage SNP chip or something?
Yes. Yes. Okay. So we wanted to determine what happened when you mutate these 2 amino acids in the TIR domain individually, just to try to confirm what we had suspected from the crystal structure. And so this is just the data I'd like to highlight here, where you can see that just by mutating that 1 amino acid that we think is involved in the protein-protein assembly, you can see an effect on the IL-33 EC50s. So this is compared to the common variant. And then you can, but of course, all 4 together has a more dramatic effect on the pathway. And then, we checked IL-1 just as a positive control. And so, okay, that was all done in a heterologous cell-based system, but what about, using actual samples from individuals that carry this haplotype? And so what we did is we have a program on campus where people can get genotyped for whatever we're working on. And so we were able to identify individuals that carried either the protective haplotype, homozygous, or the common haplotype. And I'm showing that in the blue and the red. And so what we could do is isolate eosinophils from these individuals and measure IL-33 activity. This was done by an IL-8 ELISA. And what you see here is that in red is that the individuals carrying the common haplotype, they do display IL-33 activity, as you can see here by the induction of IL-8. And then when you look at the individuals carrying the protective haplotype, you can see that it's blunted. So this is in accord with our cellular overexpression data, suggesting that this protective haplotype is somehow leading to decreased IL-33 activity. And so that's depicted here in this cartoon. So we somehow think that those amino acids mutations are affecting the downstream MyD88 cascade. And so what was also published in a paper a few years ago, that they had identified this same haplotype for something completely not related to asthma, but that individuals carrying this protective haplotype seem to have elevated expression of the soluble receptor, so this decoy receptor. And so we wanted to see whether this was the case in our own cohort. So you can see here that both at the transcriptional level in the middle, as well as plasma soluble ST2 measured here at the on the right, are both elevated in the individuals carrying the protective haplotype. And so this suggested to us that it's not just the signal that's being affected by these mutations, but it's also the level of that's somehow regulating the level of soluble ST2 to dampen the signal in vivo that way. And so that all led us to think, okay, maybe, and remember, we didn't see any association with type 2 or type biomarker low or high. So we see this as something in all asthma. And so we then wanted to try to understand a little more about IL-33 activity in vivo, and as I mentioned, it's expressed on all these different cell types. And so specifically, we wanted to know how does IL-33 affect inflammation in an infectious or allergen setting. And so the first set of experiments that I'll walk you through here is the role of IL-33 in using a helminthic infection. So this is the N. brasiliensis infection. A lot of people use it. It's an acute infection that is instigated when you infect mice with this larvae, and then you get, by day 10, you get a massive type 2 inflammation in the lung. And although it's, it's a helminthic infection, right? It's not really, an asthma model, but it does reflect a lot of the pathways we think are involved in human asthma, such as induction of IL-13, IL-4, 5, and infiltration of eosinophils into the lung tissue. And so in this set of experiments, we wanted to interrogate both the role of IL-33, but also whether there was any benefit by blocking one of these downstream cytokines in reducing type 2 inflammation. And so for this set of experiments, we used either wild-type mice or ST2 receptor deficient mice in the presence or absence of blocking antibody to anti-IL-13. So we wanted to see, okay, what's the role of IL-33 in this pathway, and can we get more of a reduction in IL-30 in type 2 cytokines if we block one of the downstream pathways? And so for the first, this first graph, this is just a fax of tissue eosinophils at day 10 post-infection, and you can see here that when you block IL-13, you have a reduction. When you have no IL-33 receptor, you have a reduction. But when you have both a blockade of IL-13 on top of your IL-33 receptor knockout, that now the eosinophil infiltration is down to that of naive mice. And that was really exciting to us because it made us think that maybe if we have more than one pathway being hit, we'll get an even greater block on type 2 inflammation. And you can see some similar in the bowel fluid, which is measured by fax. So, the hallmarks of these models is the induction of IL-5 and 13, and so we looked, this is at transcript level. You can see again that the blockade alone, IL-33 receptor deficiency alone, you have a reduction in IL-13 and IL-5, and you don't really see much when you block IL-13 in terms of IL-5 expression. So this kind of highlights that IL-33 is upstream of these different pathways. And then this is just looking at bowel fluid IL-5. Again, the combined blockade of 13 and ST2, you see a greater reduction than just ST2 alone or 13 alone. And then one of the cell types that's kind of received a lot of attention recently are these ILC2s, which are thought to be bags of type 2 cytokines. They're poised to secrete really rapidly in response to IL-33 and other factors. And you can see here that, consistent with what's been reported in the literature, when you don't have IL-33 signal, that this population is also reduced. And so we also looked at histology, so airway histology, and we looked at goblet cell hyperplasia because we know mucus is really important in lung inflammation. Sorry. And then this is just the goblet cell hyperplasia score. You can see that this particular aspect of type 2 biology seems to be modulated by IL-13. You can see here that it's completely reduced with just blocking IL-13 alone. There it is. Okay. And then that corresponds to expression of MUC5A. Oh, sorry, this is really sensitive. Okay, the MUC5A C gene, which is thought to drive mucus production in the airway. And so what I've shown you is that you have some aspects of type 2 biology that seem to benefit greatly from IL-33, blockade, such as the eosinophil infiltration. But other aspects, such as goblet cell hyperplasia, seem to be driven more by IL-13. And I'll highlight why we think that's important at the end. And so we also did one other model. This is an allergen model, which I'm sure all of you are familiar with. So it's more depicting what happens at in terms of Th2 cell biology. So in this model, we sensitize with a TNP-OVA, and then we challenge with TNP-OVA about a month later. And again, we wanted to determine whether if we just blocked, what happens when we block IL-33 alone, and what happens when we also block one of these downstream pathways. So we compared wild type versus ST2 knockouts in the presence of the 13 antibody. And what you see here is that, again, we can see that just by blocking 13 alone, you have a block in tissue eosinophil mobilization. You see this with the IL-33 knockout, and then combined again, we have a greater effect. And this is very similar to what you see in a naive mouse. A similar effect is seen here in valve fluid. And so again, just like what we saw with the acute model, which is more reflective of innate lymphocytes, in this chronic model, which has more of a Th2 component, again, blocking two different parts of the pathway seems to be more effective. And then we also looked at by antigen recall. So this is taking cells from a mediastinal lymph node and stimulating them with the antigen. And find the pointer. There it is. You can see here in the purple that combined blockade leads to a greater reduction of IL-5 and 13 production from the lymph node cells. And this is not due to any effect on cell viability, as shown here in the corner. And so finally, we also looked at airway hyperreactivity. And this is, at least in the literature, it's been thought to be driven by mucus production and MUC5AC expression. And so in the same assay, we did the same exact protocol, but this time we do AHR measurements at the terminal time point, which is day 35 here.
What, can I ask you?
Sure.
Why is airway reactivity thought to be due to mucus? You would think it would be due to smooth muscle reactivity. What's the relationship?
So we think that the mouse, that's one of the caveats of using mouse. There's just not a very big smooth muscle layer in the mouse. So yeah, I probably in higher species, that's definitely the case. There's been a lot of data with MUC5AC knockouts that show that the airway hyperreactivity has been completely lost when you don't have MUC5AC. And our data, which I'll show you, seems to correlate with that. So when you don't have 13, and in that paper actually they found it was completely driven by IL-13, at least that in that parameter. So when you block 13, you don't have MUC5AC, and then the goblet cell hyperplasia seems to be diminished. And also that correlated with the AHR.
So Bill, you've worked a lot with this model, and just comments on that?
Well, I could see how the mucus plugging small airways would be involved in the airway hyperresponse, but there's also been like the complement Marshall Willis Karp's data showing the airway hyperactivity was tied to C3 activity in the airways in the mouse.
So yeah, I mean it could just, maybe multiple factors, but with the sensitivity of the systems we're using, we're just not picking up on those subtleties. I mean, because we only did one time point, of the FEV one. I'll show you curve, and I think to see when you have multiple contributions like that, I think you have to do like a titration over time and then block the different.
Is this an invasive airway hyperresponsiveness?
I'm sorry?
An invasive airway hyperresponsiveness?
Yeah, yes.
Affects
So
again?
terminal, yeah, at the end. So one of the limitations of using this system.
You would think if it had to do with mucus hypersecretion, it would be a slow response compared to muscle spasm. Be a time course, it'd be quite different. Or don't I understand this correctly?
Well, probably. Well, I mean, you can get rapid release. From you get such a tremendous goblet cell hyperplasia. About 30% of your airways get transformed into mucus cells in these ova models. So that could be a pretty rapid response after the IL-13.
So I just wanted to highlight here is like similar to what we saw with the histology that I showed you earlier. The blocking IL-13 alone actually brings your, at least at this time point that we looked at, the FEV, the AHR responsiveness down to what you see in the naive mice right here. We didn't see an effect in the IL-33 knockouts. I think it has something to do more with the measurement that we did. Like I said, we only did one time point. It's possible that had we done a dose titration over time, we would have actually picked up on this. But what I think this does highlight, is that we appreciate that IL-33 is released. We think it's very rapid upon whatever your in allergen or helminthic challenge. But there are these other two cytokines like IL-25 and TSLP, that can also contribute and also can, provide an input to get IL-13. And so it's possible that why we see, this difference here between the IL-33 blockade versus an IL-13 blockade could be due to the contribution of these other two factors. And so that's where I'd like to now try to move the discussion into how we try to, understand the contribution of 25 and TSLP. But first, I'd like to highlight that, the one area we saw an effect when you blocked IL-33 along with IL-13 is this infiltration of eosinophils. And we know IL-5 is really important for mediating eosinophil mobilization into the lung. And so we wanted to know, okay, well, how much of the IL-5 or eosinophil response is blocked by 13, and is that why we're seeing this greater effect? That maybe together, that it leads to a diminished mobilization. And so for this experiment, we took wild-type mice and did intranasal administration of IL-33 in the presence of our anti-IL-13 antibody. And what you can see here is that when you block, you get a nice induction of IL-5, and I can tell you we faxed for eosinophils, and we also saw it correlated with eosinophil mobilization. But that is diminished when you block on top of it with IL-13, but it's not completely lost. And so we think that because of this, there's still a window that allows for IL-33 to have some effect when you have the combination treatment. And so we then wanted to do the same experiment I showed you earlier, looking at anti-helminthic infection, but this time we wanted to see what happens when you block 33 with IL-5. And so it's the same study where you infect on day 0, and we look at day 10, lung inflammation. And here we have wild-type mice or the IL-33 receptor knockouts in the presence of an anti-IL-5 neutralizing antibody. And again, we look at these various lung endpoints, primarily eosinophil mobilization. And so what you can see here is that tissue eosinophils are blocked when you, with an anti-IL-5. And I know it's hard to see here, but when you have the combination, actually, of IL-5 and IL-13, we did see it was reduced even further. Whether that's even clinically meaningful is another story because, we're already down so low. But it does suggest that combining the two pathways did provide some additional benefit. And that was just the bowel fluid eosinophils, which were also reduced. And so we then looked at this other cell type that I had described to you earlier, which are these ILC2s. They're thought to be the major source of type 2 cytokines in the lung. And we found that IL-33 is thought to regulate their expansion. And so what we did find is that this population is really not perturbed when you block IL-5. If anything, it looks a little higher. But it is blocked when you have no IL-33 receptor, which is shown here. And so this data with what I showed you with the IL-13 blockade really suggests that these upstream cytokines do control the expression of IL-5 and 13, and that it's that if you can try to block something upstream as well as downstream, that we might have more of an effect in vivo. But I'd first then like to share with you some data where we tried to interrogate how IL-25 might be also having a role in these pathways. And so here we look at what the combination of 33 and 25 is in vivo. So again, we went back to our Nipostrongylus model, and we did the same experiment that we had done with anti-13, but this time we also included an anti-IL-25 neutralizing antibody. So you can see here, if I can find, so you have wild-type mice treated with control anti-13 or anti-25, and then you have the same type of cohort with an IL-33 receptor knockout. And what I'd like to show you here is first, indeed, anti-IL-25 does reduce mobilization of eosinophils into the lung. However, and this combination of IL-33 receptor knockout with 25 on top does reduce it further, but it's not as great as what you see with an anti-IL-13. So it seems that, so this then suggests that other pathways, such as TSLP, may be contributing to IL-13 and also IL-5. And that is one reason why if we do a combined therapy, it's probably better to do one upstream and then one downstream cytokine. And so that leads me to this slide here. So what we think is that because ST2 is expressed on so many different cell types, that you get benefit from, we know from the clinic you can have some benefit when you block these type 2 cytokines. But we think that if you block something upstream, and we think because of ST2's broad expression that should probably be IL-33. And so, do you have any questions? I have a little bit more, but if we're running out of time,
No, you're fine, I think. I've got 20 minutes from what I see.
Okay.
You guys agree? Go ahead.
Okay, all right. So, so that's where we were at this point of the project. At least in type 2 inflammation, it appeared that we'd have a greater effect if we blocked an upstream cytokine and then something downstream like IL-13. But I'd then like to just talk a little bit about this other subset at the since we have some time. So we don't really understand why these patients are not responding to type 2 directed therapy. We know that by biomarker that they're eosinophil low, periostin low, phenol low. And so our current hypothesis is that this pathology is actually being driven by not inflammation, by mucus and smooth muscle contraction, hyperplasia, and hypertrophy. It is a hypothesis, and I think nobody has a really good grasp on what is molecularly going on, but that's where we're starting. And so what we think is that from data from the Brightball Lab and others have shown that in all asthma, there is an infiltration of mast cells and smooth muscle. So that's actually where we've kind of been focusing our research, is to understand the mast cell smooth muscle communication as a way to try to understand this type of asthma that's not responding to type 2 directed therapy. And so ST2 receptor is expressed on mast cells, and that was another reason why we were interested in this pathway. And it's been shown in the literature, and I'll share with you some of our internal data, that IL-33 can potently activate mast cells. And so the first set of experiments, and this was published by others, and we just repeated it in-house, is that in the presence of IgE, anti-IgE, so these are in vitro human mast cells that we derive from PBMCs. So you culture them for about 2 months, and you get mast cells, and then you can sensitize with IgE and challenge with anti-IgE and measure degranulation of various transmitters. And we did this in the presence or absence of IL-33 to ask whether IL-33 could augment mast cell degranulation. And what we found is that indeed, and this is exactly what was published also, that you have release of beta-hexaminidase, tryptase, and histamine with the anti-IgE, IgE cross-linking. This is increased when you add IL-33 to your cocktail, and then this is just our positive control, which is the calcium ion. And so it looks, at least in vitro, that IL-33 can augment IgE-mediated mast cell degranulation. What we also found, and that others have reported, is that IL-33 can directly activate mast cells to secrete cytokines, either in the presence of the IgE crosslinker, and then I'll show you in a minute without. So here we looked, instead of an immediate examination of degranulation, we looked at cytokine secretion in response to IgE crosslinking. So this is 3-day cultures. You can see that IL-8 is secreted, but only in the presence of IL-33 do you see induction of cytokines such as IL-5 and IL-13. And again, this was also reported by others.
These are human cells.
These are human cord blood cord blood derived, and we expanded them for 2 months with different factors. So it's a little tricky to measure mast cell biology, especially, of course, in the lung of a mouse, just because of the anatomy. But there are ways to look at mast cell mediated or IgE-mediated anaphylaxis in vivo. And so you can take mice and provide the anti-DNP, which is your sensitization, and then challenge with DNP-HSA. And then you can measure anaphylaxis as a change in basal body temperature over a period of an hour, and this is done in increments of 5 minutes. And what we did in this experiment is we wanted to see whether adding IL-33 could augment this systemic anaphylaxis response. So kind of a surrogate of what we showed you with the cell-based assay in terms of mast cell activity. And so we did this, and just show you in the triangle here, wild-type mice treated with the IgA, cross-linking with IL-33. You can see this change in basal body temperature. And this is affected when you add So first look at the knockout mice where they respond fine. This is shown in the literature, so you can have an anaphylactic reaction. But when you add IL-33, of course, nothing happens. But when you take wild-type mice and then you do the same experiment and you've added IL-33, similar to what we showed in the cellular cultures, that it does seem to increase the anaphylactic response as measured by a change in basal body temperature here. And you can see, find the pointer. All right, so you can see here this was measured. We did an area under curve measurement as well. And so, of course, all of this, it's not lung mast cells, so we can't, predict that this is going to happen there. But it does imply that IL-33 can augment many of the functions of IgE in vivo. And so there was also one other report that IL-33 could affect mast cell viability. And so for this set of experiments, we took mast cells and either used SCF, which we know is a mast cell survival factor, or IL-33, and we looked at viability. And it was similar to what was published in the literature, is that IL-33 can, this is a measurement of apoptosis, so an Xn5 staining by FACS. You can see here that when you don't have SCF in the control, you have high apoptosis. However, this is reduced in the presence of IL-33. These are 3 different donors. There's really nothing different about them. And this is various degrees of increasing viability. We also looked at percent viability, and you can see here that compared to control cultures where we removed SCSF, that by adding IL-33, you now have some increase in preventing cell death. And so, where does this
Can I
lead
ask you a
us?
question?
Sure.
Based on that, is it thought that IL-33 may play a role in systemic mastocytosis?
Yeah, that's not been investigated, actually. Good point.
I think systemic mastocytosis is downstream, right? Because the mutations would be,
Well, the mutations would be separate from this totally, but this would keep cells viable and maybe totally unrelated. Just struck me to ask that question.
So finally, where I'm going with all of this. So we appreciate that targeting, type 2 cytokines has an effect in these eosinophil high patients. And we've really tried to understand how we can do better. And that led us to the IL-33, trying to look at IL-33. So I showed you genetic data that there are SNPs that associate with protection from asthma risk. And then we know that IL-33 has multiple effects on these different cell types. And when we combine blocking IL-33 with, say, IL-13, one of these downstream mediators, we're actually able to get a greater effect on type 2 inflammation. We think it's because, we're blocking the IL-33 signal, which is, activating these different cell types, but by blocking a downstream cytokine, we're also taking care of the effects of TSLP and IL-25 and other factors that might be regulating these downstream pathways. And then, I know it's all cell culture and it's a lot of, hypothesis right now, but we think that this effect of IL-33 on mast cells may actually have some role in what modulating, mast cell activity that might have a role in the pathogenesis of this type 2 low or inflammation low asthma. And so that's an area where we're really trying to understand better with, better animal models, because the mouse is actually a really bad model to look at mast cell in the lung. But to try to understand how IL-33 might be regulating mast cell function, specifically, smooth muscle activity. And so that's where we are. We're trying to understand how blocking 2 pathways could be better than blocking 1 alone. And then that's just my acknowledgement. acknowledgement. So I know I ended early. I'll be happy to go back and whatever was missed and elaborate more. Go ahead.
So it's some very nice data, and I guess one broad question, if you think about translating this into a clinical setting, I mean, who's going to do a study of dual blockade? You know, drug companies and the FDA are very hesitant, I know, to combine biologics.
Yeah, yes. You're right. So the original idea was like try to do like a bispecific. Right now, we're actually just interrogating ST2, the IL-33 receptor, and the idea is we'll see how that plays out. We're starting the trials, and then maybe you know everything I showed you was in mouse, and of course when you're using the fax machine, you can always see like subtle quantitative differences, but maybe just hitting upstream is enough to see an effect. I think that's the ultimate experiment is in the clinic to see is hitting upstream enough to see these changes in these. This is a really sensitive. Anyway, these changes in these parameters that you guys you measure in the clinic, or are we going to have to hit harder because of cytokines such as IL-25 and TSLP that might be contributing as much? And I think we just can't tell.
I mean, it makes a lot of sense to use the combination therapy, but is there any model out there, or is any drug company doing that in any disease?
No, I mean, Dupy is sort of combination. I mean, they are going after the IL-4 receptor, but they're in theory hitting two cytokines.
Okay.
And then we are also doing totally unrelated to this, but we have an IL-13/17 program that we think will hit. It's a bispecific.
Yeah.
But we think that's going after a different biology.
Sure.
So, go ahead.
So are you suggesting maybe instead of two antibodies actually having soluble receptors for 33 plus, antibodies to the other, instead of two antibodies, because there's that issue with like Remicade and Xolair at the same time.
Yes, I know. Soluble, there's already so much soluble ST2 at baseline.
Gotcha.
It would be really hard to dose over that, I think. Yeah.
For the IL-33 study that you're proposing, we were approached to do it here. That is anti-IL-33?
Receptor.
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 But not soluble receptor blockade.
Right. So our receptor program is going to start phase 2 soon.
That's a monoclonal or a small molecule?
No, it's a monoclonal to the to ST2. It's basically anti-ST2.
With an N of 1, I wanted to put a patient on Xolair and Kala, but they didn't let me. So.
Yeah, we have one patient on both who's doing really, really well.
I would think it'd be great, the concept here of hitting two different targets. So, I mean, your program for basically anti-IL-13 didn't make its endpoint, right?
Connie can elaborate more. I'm not on the 13 team.
Yeah.
So not in asthma, at least not clinically. So one of the phase 3 trials did not meet its primary endpoint.
And that was in the periostin high?
So periostin high and periostin low.
You would predict it in periostin low.
Exactly.
That's not a surprise.
But then even in periostin high, in level 2, which is one of the phase 3 trials, there was no statistically significant reduction in asthma.
So is that program going forward?
No, so the asthma program is not going forward. But the lebrikizumab, atopic derm, and the COPD program is still ongoing.
Patients that have the SNPs associated with higher risk of asthma, is that a gain of function mutation they have? Increased expression, secretion of IL-33?
So we think it's a protective mutation, and so we think it's actually protecting from asthma risk, and it seems to be some kind, not loss of function, but subtle change in activity.
No, I meant the other group, though, that has been associated with. Is there any gain of mutation that's been described?
No, we haven't found that.
But what about Fahi's data showing severe asthma associated with like increased IL-33?
Oh, yeah.
Airways
No. And many people, we've tried, the IL-33, we did it by gene signature, and it's really hard because it's NF-kappa B, so everything lights up. But yeah, we worked with John Fahy on some of his stuff. And it does definitely look like there's more IL-33 in the severe asthma. If you look at, some people have even had, Dr. Hamid did some quantification by IHC, and you could actually see increased smooth muscle expression of IL-33. But it's been hard for us to translate that expression to activity just because this NF-kappa B is the major signal, and everything signals through NF-kappa B. So as a transcriptional activity anyway, we haven't been able to say, okay, this is due to IL-33, make that connection.
And has there been anything found out about that group of patients that seem to they fit in the high TH2?
Yeah, no, not at all. And most of our data so far suggests that it's just looking at soluble ST2 expression in these patients and not segregating by biomarker. We seem to see it in both, like low and high, and same with the SNP, right? We see low and high, which is why we think it may have a role even in the low. I mean, we'll do the clinical experiment and we'll actually see for sure. But how it's having a role, it could be mast cells, it could be some other cell. We don't know yet. But that's a good question. I mean, when you think of IL-33, though, you think of TH2 biology. I mean, that's the main driver. So we would predict that that's where we'd see the signal when we do the trial, but we don't know yet.
In your viral-induced models, where you have RSV or something, that would be as good a trigger. So that would be the non-TH2 phenotype that would, or pollutant that would trigger the airway epithelial damage.
Yeah, so I know that Dr. Holtzman has tried a lot of the viral models, and I think Dale Matsu, before he came to Genentech, he was trying a lot of those and published on it. Yes, and they seem to think that that's IL-33 driven. And what we know about this TH2 low, we think that it might be having a viral component also. So it's very likely maybe that's how IL-33 is contributing. Hard to say at this point.
I was texted a question here from the outside, which is a very good question, I think. Is the expression of IL-33 steroid responsive? And then, actually, it leads me to think, just old-fashioned clinical medicine, is any of this better than steroids? Except for the safety issue, does any targeted therapy like this work better than prednisone?
So the cellular data, there's been conflicting reports. We actually did some studies with steroids and saw some IL-33 responses were not affected. We did it transcriptionally. We just compared what happens with and without steroid on top with IL-33. And some pathways seem to not be steroid sensitive. But I know there's data in the literature that's been published that has thought that most of the 33 signal is steroid sensitive, at least in the mouse model. So, it's hard to say at this point. Yes?
Is there any other side effects of these knockout, like if you knock out IL-25 or knock out IL-33, is there any other derogatory effects on these animals? Or are they just normal, happy animals until you insult them with a parasite?
They're normal, happy until you insult them with anything. But, I mean, there's, you know how it is with mouse experiments. You give the right dose, look at the right parameter, you'll see an effect. So people have published IL-33 has a role in CD8 viral responses. We looked at the data. It was an LCMV infection. They gave a lot of virus. So how do you interpret that? I don't know. There's Fiona Powery has a lot of data on IL-33 in the gut, on saying that its role on Tregs. That if you perturb IL-33 activity in the gut, you get like colitis. It's a protective effect. Not everybody's been able to reproduce that. I think that the IBD models are just tough with the whole flora issue. And it may have a role in Tregs in the gut, but then we also have learned from, the psoriasis studies with like IL-17 that you can have a protective effect in the gut and have a pathogenic effect in the skin, and it's just like a location-dependent effect. So we don't know if blocking 33 in the lung may be fine, and then if you block it in the gut, it may not be fine. So, all of this is we have to figure out. But
And kind of follow up to that, have you looked at any other models with like a dust mite model?
Yes, they're the same. We see the same thing. No effect on neutrophils because the dust mite model has a neutrophil component to it, but we do see the effect on eosinophils. So we looked at dust mite, we looked at ozone, there's aspergillus, triple amyloid. They all eosinophil models seem to have an effect when you block IL-33.
Does 3325 or TSLP have any autoregulatory effects on the epithelium itself, or is it all going to the inflammatory system?
So we have not been able to show effects of either 25 or 33 on epithelium, like in our lab. Others have published it, but we just cannot reproduce any of that data. And as far as TSLP, that was also published in the gut. we tried to look at that, and it didn't. At least in the gut epithelium, we couldn't see any TSLP receptor like actual activity, but who knows?
Any other questions?
And I apologize for no, now I've actually mastered how to use the pointer, and by the end of the presentation.
So come back next week and we'll have you again.
That's right.
You talked to Steve lately?
Yeah, you go.
Sorry we gave you a difficult computer.
You know, when I was at IACI, it was the same thing. I'm just not used to we always like have the I don't know how much actual things are out there. But IACI, it was the same thing, and I remember struggling there.
The only reason my phone's telling me my
What? Just for
Correct. Yeah, they can't see what they do.
It's somebody who's just.
No worries. I hope it was clear.
Again, oh, where are you now?
Former fellow colleagues.
I don't know how to make
Okay, so what's your main
How are they doing?
They're good. Like the little
a lot of discussion, which I like. I mean, it's a very, very proud.
I'm not the one to make
Because like I just saw the only source of change, but the hours. Oh, I said yes. But we left about 30 minutes after whatever presentation. So it's fun though. New company. Then we have
Yeah, I'm not on the clinical team. I handed it over and I don't have access directly here.
And they've got a new person coming too.
Yeah, no, I'm sorry. I'm just trying to meet everyone. Going to CIS.
It's a done deal or not.
Yeah, because your name is on the agenda and you are the primary investigator.
I am. I don't know if you may not be going along. First way would be to see if your criteria were something that couldn't
I haven't used it yet.
let me know when I get back. Sure, I'd like to.
I mean, I said,
So the investigator meeting